Just double checking, Tatiana, that I pronounce your name correctly. Is it Tatiana Giraud? Is that right?
Yeah.
Okay, great.
Sure. And I'm sorry, I didn't pay attention, but our university again changed name, so now it's Paris-Saclay, and not Paris-Sud anymore, but just
Paris-Saclay?
yeah, Paris-Saclay. Sorry about that, but it doesn't matter.
Okay, good morning, everyone, and welcome to this seminar series that is organized by the Special Topic Network, Integration of Speciation Research. And I would like to highlight that there's one more seminar that is scheduled for this year, and it is December 5th, with the theme Macroecological Perspectives on Speciation. And the speakers have been confirmed. We have Katie Wagner and Daniel Cadena. And just a little bit about the seminars, how they work. So the first hour is dedicated to the two talks. These are recorded and posted on YouTube, so you can check them out later if you miss. And after that, the last 30 minutes of discussion is dedicated to the theme of the seminar, where you can contribute by posting questions to the chat. And you can also post questions about the specific talks during the talks, and we will try to ask them after the talk if there's time. But before we go to the seminars of today, I would like to briefly highlight an event that is of interest to this community. And it's an event organized by Linnean Society, Perspectives on Speciation, and it's April next year. And the aims of this meeting is very much also what we are also aiming at in IOS, which is to share perspectives, encourage communication across disciplinary divides. So please check this out. But now let's go to the theme of today, which is species interactions and speciation. And Tatiana will go first, and I'll give the floor to Joanna to introduce Tatiana.
Hey everyone, it's my great pleasure to introduce Tatiana Giraud. Tatiana is a researcher at the CNRS University Paris-Saclay. She's a member of the French Academy of Sciences, and she does really amazing research on fungal pathogen speciation in those combining theory, comparative analysis, population genomics, and experiments. So I very much look forward to your talk. Tatiana, please, the stage is yours.
Thank you very much, Joanna, and thanks the organizer for the invitation. So I'll share slide. Can you see it? Perfect. So yeah. I'll speak today about the work we have been doing for several years on the speciation in pathogenic fungi, and try to explain some of the features that we think are conducive for sympatric speciation in pathogenic fungi. You can see here one of the pathogens we are working on. So here in the anthers of the Saline latifolia flower, you can see spore of a fungus, the anther smut fungus, that castrate the plants and parasitize them. And so they kind of hijack the reproductive system of the plants by producing their own spores in the anther of the flowers that are then dispersed by pollinators. And that's one of our main model, but we work also on other fungi, other pathogenic, also mushroom. And so one of our question is trying to understand how species are formed in fungi and whether there are some specific aspects on pathogenic fungi or mushrooms. So when, as you know, when we work on speciation, one of the main questions is how does reproductive isolation evolve? That's one of the main issues in speciation, to understand how two lineage can diverge, and that's mean how you can have assortative mating, how you can stop gene flow between these two incipient lineage to avoid producing unfit hybrids. And so one of the most terminal and first work on how reproductive isolation evolves is that from Coyne and Orr, where they assemble data from pre-zygotic and post-zygotic isolation in Drosophila, and as a function of genetic distance and depending on whether the species pair are in allopatry and sympatry. And they found a very interesting pattern where in allopatry, both the pre-zygotic and post-zygotic isolation gradually evolves. It's just a byproduct of genetic divergence. While when species pair are in sympatry, reproductive isolation, it's pre-zygotic here. Pre-zygotic evolution, isolation evolve very rapidly and is already very high, even at very low genetic distance. And the interpretation is kind of like there is a selection for pre-zygotic isolation when in sympatry to avoid producing unfit hybrids. So we ask whether we have the same pattern in fungi. And first we look at basidiomycetes, so that one of the two main groups of fungi that include the mushrooms, but also the rust and the smut. You have here the corn smut fungus producing tumors in maize. So the grain become bigger and full of spores of the fungus, and actually that's very good to eat. It's huitlacoche in Mexico. And so in this group, we kind of found the same pattern as in Drosophila with the pre-mating reproductive isolation increasing gradually with genetic distance, so with time when in allopatry, while when the species pair are in sympatry, there is a pre-mating reproductive isolation that is very high, even at very small genetic distance, which can be interpreted as selection to avoid producing unfit hybrids. So for instance, if you imagine fungi that parasitize these two host species, these trees, and that you need to be little a and little b to infect this tree, and you need to be big A, big B to parasite these three species. But if you mate in sympatry, regardless of your genotype, you will produce intermediate that are neither able to infect either host. So there can be a selection to avoid producing these intermediate hybrids. But surprisingly, in ascomycetes, so the other group of fungi that include the molds and actually most of pathogenic fungi, like here the botrytis on grapes, the Venturia, the apple scab fungus, or these Ascosita fungi parasiting these crops, and also all the molds, ascomycetes. And here we didn't find these reproductive character displacements where there is a difference between allopatry and sympatry, and very surprisingly, even in sympatry, pre-mating reproductive isolation could be very low, but also at relatively high genetic distance. So I think that surprisingly, there is no selection to avoid producing hybrids in sympatry, and that even sympatric species can persist without pre-mating reproductive isolation. So that's data collected from the literature where mycologists try to cross in vitro many kind of fungal species. So that was kind of surprising, the difference between the two groups and this pre-mating reproductive isolation. But actually it can be understood, and we did a kind of modeling to explain that. And one particularity of this ascomycete fungi is that they mate after having grown in their host, so or in their habitats. So the situation is very different from the basidiomycete or most animals or plants, in that so they first parasitize their host, they grow inside, and then, for instance, this apple scab fungus, it parasitize in the spring and the summer, the leaves, the apples, and then in the fall, they do the sexual reproduction within the leaves that have fallen. So it's only the individual that have been able to parasitize a given host individual that can mate together. So actually, that's kind of already a pre-mating reproductive isolation. There is kind of a pleiotropy between the ability to infect a given host and then the probability to mate. So they first parasitize their host, and then they mate within the host. So they can only mate with other individuals that have been able to infect the same host. So if you think that there is a very strong selection on given genotypes to infect a given host, for instance, here only the big A, big B can infect and be in this host, they can only mate with big A and big B without evolving any active pre-mating reproductive isolation. And that kind of the same concept as a reduced viability of immigrants. You have a strong selection to be able to persist in a given habitat and then mate there. And actually, there could be other feature of this pathogenic ascomycete that are conducive to sympatric speciation. So there's kind of pleiotropy between host adaptation and mating probability. And there is also a very, very strong selection, and on one or two genes. There is this so-called gene-for-gene relationship in plant fungi interaction, where there are some resistant genes in plants that produce protein recognizing molecules from the pathogen. And when the resistant the protein coding for resistance recognize the pathogen, it completely blocks infection by killing its own cell around the pathogen. So the pathogen cannot enter, it cannot get resources. While if either the pathogen has another protein, or the resistant protein does not recognize the pathogen, then there is full infection. So it's really a given one gene that code for something like all or nothing in terms of fitness of the pathogens. So if the plant has a resistant gene, it's just zero fitness. So it's very, very strong selection, and so it can really prevent mating within the host between fungal individuals that do not carry the same alleles at just one gene. And another feature that may be conducing to sympatric speciation is that you can see here a wheat field parasitized by the rust fungus, and you can see here all the spore of the rust fungus dispersing in the air. So it's like billions of spores, and that makes huge opportunities for new novel mutation to be able to infect the new genotypes, new host species. And it can also allow for this huge selection load imposed by the very strong selection to infect or not the host. And so there are many, so all ascomycete, and in particular pathogenic ascomycete, made within their host after having grown and be able to parasitize them. But it's the same thing from saprophytic ascomycete or others. They have to first grow in their environment and then mate within the habitats. And we actually studied kind of almost sympatric divergence directly in this apple scab fungus, Venturia iniculis. So there was so this disease is very problematic because it makes apples that people do not want to eat. So there are lots of fungicides, which is not good. So people try to breed resistant trees. And actually, so in France, there was a resistant genotype that was very successful. And for 40 years, there was no disease on it. But eventually, there was a population that could break down the resistance of the tree and parasitize the resistant trees. And you had in orchards, so in sympatry, in the same orchards, you had resistant trees and susceptible trees. And on the susceptible trees, you had only the ancient population of this fungus able to parasitize only the non-resistant trees. And you had a new population on resistant trees that was able to infect these trees despite their resistance, but that was not able to infect the susceptible trees. So you had like the whole specific population of fungi, specific of genotypes of trees in sympatry. They could only infect their own genotypes, go there and mate within their host. We followed the population for 10 years and there was no detectable gene flow, despite the fact that in the lab we could cross the two populations without any issue. There was no pre-mating isolation, no hybrid invariability or infertility. But in the orchards, in sympatry, there was no gene flow for 10 years that we followed because of this pleiotropy between the ability to infect the host and the probability to mate. And so I showed you this pattern of reproductive isolation evolution in the basidiomycetes. And so the pattern was like classically found in animals. But there was this strange point here. So species in sympatry with low pre-mating reproductive isolation. And that's precisely one of our favorite fungus, the Antersmut fungus. So we try to study more their reproductive isolation evolution. So this fungus, you can see here in the Salini latifolia species that is dioecious. So you have female plants and male plants. But in both males and females, the fungus is able to grow, to make them grow anthers full of spores. So it cannot transform even the females in phenotypic males bearing anthers full of spores. And actually, you have here a drawing by Charles Darwin of this transformation of female flowers into males, male-like flower with anthers that Darwin wrote in a letter. So he had a correspondence with this Lydia Baker. She was a botanist, very good and feminist. So she was one of the first to advocate for that the women should be able to vote. And she wrote to Darwin about this fungus, saying that there could be that there's no that much of difference between males and females and men and women because even a fungus can transform females into a male. And so you have here the life cycle of the fungus, where diploid spores are produced in the anthers of the plants, transmitted to healthy plants by pollinators. And so when the diploid spore land on a new host plant, it can germinate by undergoing meiosis. And you have most often intratetrad mating, so mating within product of the same meiosis, and then a dikaryotic hyphae grow, infect the plant, and then the next flower that are produced are diseased, full of spores of the fungus. And so we try to understand the coevolution, co-divergence between the Antersmut and the plants. And one cool thing is that you have this Antersmut disease in many host plant species of the Caryophyllaceae family. You can hear on Dianthus, Salinis, and other kind of Caryophyllaceae plants. And so one of the first questions was, for instance, whether you had co-divergence between the fungus and the plants, or whether there was a lot of host shift with the parasitize the pathogen, forming new species by shifting host on extant plant species. So we try to reconstruct the phylogeny of the plants and the fungus and try to see whether it was congruent, which indicates usually co-speciation. And so you have here the phylogeny of the microbotrium pathogen, and here the phylogeny of the plants. And you can see that not congruent at all. There have been a lot of host shift. And actually, we did some kind of literature survey, and it's really the rule actually in host parasite interaction, in contrast to the idea that was long in the literature that coevolution should lead to co-speciation. Actually, between host and parasite, there is almost never congruence between the host and pathogen phylogeny. It's mostly divergent by host shift in pathogen interaction. And so then we try to understand how there could be lack of gene flow between these pathogen species. And so trying to cross different species and trying to infect plants and see how it changed with genetic distance. So we tried first to see whether there were pre-mating isolation. So you can see here the kind of gametes of the fungus, and you can have mating only between two different mating types. So we isolated one mating type from one species and the second mating type from the other species, and tried to cross them by seeing whether they would grow conjugation tubes. And you can see here the proportion of conjugations as a function of genetic distance between the species. And there is no change in the proportion, even so they are able to make the conjugation tubes regardless of the genetic distance between the species. So no evolution of pre-mating reproductive isolation. And then we look at the post-mating isolation. So first, the ability to grow this hyphae after conjugations. And you can see here the inability to produce the hyphae. And here, it increased with genetic distance. So the more distant the species, the less able they are to make a hybrid hyphae. And then we try to, make the hybrid cause disease, so inoculating the plant in the greenhouse and then looking whether they would produce symptoms and they're full of spores. And you can see here also that there was also a strong post-mating isolation. So you have here the complete, with one is the complete inability to produce symptoms. And so it arrives rapidly at small genetic distance, the complete inability of hybrids to infect the host. And the rare hybrids that are able to infect the host, actually they produce like aberrant symptoms. You can see here hybrids that did not completely prevent the production of pollens in anthers. So there are a bit of spores, but also still pollen. Or here, completely abnormal symptom with the production of spores in the ovules of the plants, which never happens in the microbotrium fungi. But which is interesting is that actually some other smut fungi do produce spores in the flowers, in ovules, and so it may be like some ancestral traits coming back in hybrids. We also look at the evolution of hybrid sterility, and it also increased with genetic distance. So we look at the ability of hybrids to conjugate and so trying to mate. So it also increased with genetic distance between the species. And interestingly, the genome size in the hybrids increased also with genetic distance. So it looked like there are some abnormal meiosis and abnormal genome content in the hybrid. So maybe linked to chromosomal rearrangement between species, and so meiosis that does not work very well. And actually, we found that there have been a striking evolution of the mating type chromosome in these fungi. So you can see here a phylogeny of this microbotrium fungi on different host species. And you can see like the ancestral mating type chromosome of this fungi. So usually basidiomycete fungi have 2 mating type loci that we call Pierre and HD loci. And in this Microbotium species, it occurred repeatedly. All the arrows here is an independent event of linkage of these 2 mating type loci on the same chromosome by chromosomal rearrangement, but by different kind of chromosomal rearrangement. In some species, you had the 2 whole chromosome fused together. In other, it was only 2 arms of the chromosome. In other, it was 1 chromosome fused with another arm. But that means that in these different species, you have completely different mating type chromosome with different gene content. And so obviously, it cannot work very well at meiosis. So that may be what kind of explanation of this strong post-mating isolation, and in particular, the hybrid sterility and abnormal gene content in the hybrids. It is huge flexibility, fluidity in the genomic rearrangement, and in particular on the mating type chromosomes. But still, the question remains. So we don't have pre-mating isolation, apparently. And it's not like in ascomycete, where the host adaptation control also the probability of mating, because in this fungus, mating occurs before plant infection. So there is no pre-mating isolation in sympatry, but there is strong hybrid sterility and inviability. So we could think that there should be a selection to increase pre-mating isolation, or we should see more hybrids. Or there is still a question here. But we thought maybe in this fungus, another trait conducing for sympatric isolation is that they undergo a lot of selfing, and they deposit a lot of spores on the plants. So you have here, for instance, one species parasiting this Lychnis floscuculi plants, and here another Microbotium species parasiting this Silene latifolia plant. And so in the rare case where the spores of the 2 species are deposited together on the new plants, actually, so there are lots of spores undergoing selfing, and some other small fraction undergoing hybridization. But you have systematic competition before entering the plant between hybrids and non-hybrids, right? Because you always have selfing on the plant. So even if the self intraspecific force has a small advantage, finally, eventually, it will be this one who infect the plant. So even a small advantage actually transforms into a huge and all or nothing advantage because of this systematic competition between hybrids and non-hybrids before even producing a hybrid individual. And which we performed an experiment to test that. So we infected hybrids alone on a plant or in competition with non-hybrids. And when the hybrids were in competition with non-hybrids, they almost never actually succeeded in infecting the plant, while when they were alone on a plant, sometimes they could produce diseased plants. So there is like a competition arena between hybrids and non-hybrids that could actually make that you don't need to evolve pre-mating isolation because it's a byproduct of this competition arena. Then we studied more precise. Oh, sorry, the slide are not shown very well. So you have here the phylogeny of this Microbotium fungi, and we studied these 2 sister species, Silene latifolia and Silene dioica, to try to understand the speciation history in this species pair. So we collected fungi on these 2 host species. And so with either microsat or full genome, we looked whether there were some spillover on hybrids. So it looked that there are some spillover sometimes, but very few hybrids. And actually, by sequencing full genome, we didn't detect hybridization at all. And so we try to infer the history of gene flow. And so you have here the inferred gene flow in the 2 directions from the past to the present. And what was striking is that at the time of speciation, there was no gene flow at all, but increasing toward the present. So that make think of like allopatric speciation and secondary contact, like something like 400,000 years ago. And then we studied the other two sister species that were very interesting because based on single gene genealogy, we could not differentiate them. And so that's one of the very rare native North American Silene species. And so we thought maybe that one of the few generalist microbotium species and these two hosts that are also closely related. So we sequenced a whole genome on this Silene virginica host species and on 3 subspecies of this Silene caroliniana species. And actually with whole genome, we found 3 microbotium species, one specific of Silene virginica and also of one of the subspecies of Silene caroliniana, and 2 other clusters, each specific of one subspecies of Silene carolina. So it looks like you have already host specialization even at the subspecies level in these plants that are, despite the fact that this plant subspecies is very hard to distinguish. You need to really be a specialist, but the fungus knows. And we try to infer the history of gene flow. And here it was actually the opposite. We inferred that the speciation occurred in the face of gene flow, but that gene flow decreased with time, and that and now there is no more gene flow between the 2 main clusters here. And we also infer that the gene flows was heterogeneous along the genome. And actually, we found 2 large inversions between these microbotium species. You can see here the genome with different chromosomes, and in blue it's when they are synthetic, and in orange when there is inversion. And so there was 2 inversion on 2 chromosomes, 2 large inversion. And strikingly, there was a lot of FST divergent outliers and positive selection outlier here, precisely on this inversion. So it reminds a bit of the history of Rhagoletis or other speciation where actually in sympatry, big inversion allows to keep the suite of co-adapted alleles together that could allow to infect these 2 different host species. And so at the conclusion, we found some traits in pathogenic fungi that could be indeed conducive to sympatric speciation, such as the mating within host that induce a pleiotropy between host adaptation and mate choice, very strong selection and with billions of spores, and also this selfing and competition arena situation that also, and that may explain why there are that many pathogenic fungi specialists of different plant species. Could be, of course, also extended to other host pathogen system. And with that, I'd like to thank a lot of students, postdocs, and collaborators and funders. And I thank you for your attention.
Wow, thanks very much for this amazing talk, Tatiana. What an amazing example of species interactions being very important for speciation. If you have any questions, anyone, please just post them in the chat as Jonna has written already. Given that there's none yet, I will just start. Tatiana, how do you explain the lack of similarity between the phylogenies of the host plant and the fungi? Is it because there's very, very commonly switches between hosts?
Yeah.
Or
yeah.
is it, okay. And do you usually find that the fungal phylogenies suggest that the fungi have speciated more recently than the host plants?
Yeah.
Or have they speciated more
Yeah.
anciently and then just switched around when there was opportunity?
Yeah, the time in the nodes are much more recent, the fungal phylogeny than the host phylogeny.
Okay.
I have a follow-up question or related to this. I was wondering whether you have any evidence of hybrid speciation, and especially related to the host shifts. So does that occur in this system?
Example of what, sorry?
Hybrid speciation.
Oh.
Hybridization,
yeah. So we didn't find any evidence of that in our fungi, but there are examples in pathogenic fungi where there have been a hybrid speciation, for instance. So pathogenic fungi are mostly studied in crop forest trees cases, for instance. So people also try to grow resistant poplars that would be resistant to rust fungi. And so they found that when hybrids between two poplar species were resistant to the rust of either parental species, but then a few years later, actually the fungi hybridized and were able to infect the hybrid trees, and they mate within their host and all.
So it was sort of co-hybrid speciation.
Exactly.
Interesting.
And there are also other examples. So in more natural system, like there are some pathogenic of grasses, the epicloe. They also sterilize their host by producing their own sexual structure in the place of the sexual structure of the plants. But there are hybrid species that are asexual. And they actually became symbiotic of the plants. So they do not produce sexual structure anymore. They stay as endophytes in the plant, and they even produce molecules to help the plants fight against the herbivores. And so they are generating a new hybrid asexual species and with completely different lifestyle, shifting from parasitic to actually beneficial, symbiotic to the grasses.
Guys, there is now a question from Claire Merot in the chat. Can you see that, or I can also read it?
Yeah. So the question is whether the larger genome size in the hybrids is linked to more transposable elements or doubling of rearranged section. So we didn't check actually in hybrids, we didn't sequence the hybrid genome, but because later we found that these mating type chromosomes were completely rearranged, what I think is that so if you have just basic segregation of the mating type chromosome, you will, because they don't have the same gene content between the species, you will lack a lot of gene and being inviable. So the only hybrid that can copy that, for instance, kept two mating type chromosomes from the two species, and so they have a higher gene content because they kept the mating type chromosome from the two. They didn't just kept one. That has a very high amount that would be that rapid. It's the chromosomes. Thank you.
Thanks. So maybe in the interest of time, we move on to the next talk. So it's my great pleasure to introduce a colleague, Astrid Groot, who is the head of the Evolution and Population Biology Department at the University of Amsterdam and the outgoing past president of ESEB. Her central research question is how sexual communication affects speciation, and she investigates the species interactions from predator-prey to plant-insect interactions and how they impact sexual signals and responses within and between populations. So the floor is yours, Astrid.
Thank you very much for this nice introduction, Jonna, and also for this invitation. I'm very happy to be here and to talk about our work. Let me see, here is my... So can everybody see this? Okay, perfect. I will talk about how or does interspecific communication interference affect speciation. And if you think about communication interference, then it's about reproductive character displacement, where two populations evolved similar characters in allopatry, and then upon secondary contact, there is selection to diverge in these characters to minimize reproductive interactions. But what if, oh, does this not work? Yeah, what if the trait, this character, is costly? And what if after in this hybrid zone, or in this overlapping zone, there is evolution towards niche differentiation or host differentiation, the example that Tatiana, for example, talked about. And then we have non-overlapping areas in these overlapping areas. And if the trait is costly, could it then lead to the evolution of trait loss, maybe? Well, I'm focusing my research already for more than 20 years on the evolution of sexual communication in moths, and where the females produce a species-specific sex pheromone in this sex pheromone gland. So this is a little video, and she is actively extruding this gland from which the pheromone blend is extruded, emitted, and then towards males are responding. And we know a lot about these sex pheromones because moths are important pests in orchards, and we can use these pheromones for monitoring and mating disruption. And what I'm trying to depict here is that every species has their own species-specific sex pheromone blend, but the pheromone components are similar across the different species, and the species specificity lies in the presence-absence of certain components and the ratio of these components. And so we also know a lot about the sex pheromone productions. So these sex pheromones are produced in a certain pathway, but from fatty acids. They come from fatty acid synthesis that is all happening in this sex pheromone gland. And then the final products are aldehydes, alcohols, and acetate esters. And biosynthetically, we also know what type of enzymes are involved. And so today I would like to focus on the acetates, the acetate esters, but I will abbreviate those to acetates. And most of the moth species produce these acetates. There are only a few species that do not produce these acetates. And when we focus on two specific species that I have worked on since my postdoc, I started as a postdoc to work on these species, this Heliothus virescens and Heliothus subflexa. By now they are called Chloridea, but I'm sorry, I have worked for this with these species for more than 20 years, and I just can't get it into my head, this new name. So I will keep referring to Heliothus virescens and Heliothus subflexa. These two closely related species co-occur throughout North and South America. They diverged about 2.5 million years ago. And the main difference in their sex pheromone composition is the presence or absence of these acetate esters, these acetates. So Heliothus subflexa produces these acetate esters, while Heliothus virescens do not have any of these. However, you would think that if we want to then know what is the difference between these two species in terms of genes, then we would just do a genomic analysis. But it's not that simple, because the absence of acetate esters is tissue specific. So even though Heliothus virescens females do not have this in their sex pheromone gland, we know that these acetate esters are present in the male hair pencils. So at close range, males also produce a sex pheromone from their hair pencils, and their acetate esters are present. And also, Naomi Sweres, a PhD in my lab, has found that acetate esters are also present on the legs of males and females. So it's so genomic comparisons is just uninformative. But what I did do when I started in the labs of Fred Gould and Kobe Schall as a postdoc, we did QTL analysis, quantitative trait locus analysis, to identify the regions that underlie this variation. And what we found is two chromosomes. So these moths have 31 chromosomes. And by doing this QTL analysis, we found two loci, chromosome 20 and chromosome 28, that specifically significantly affect these acetates. So if the heterozygote females produce significantly less acetates than the homozygous subflexa females. Now, we first wanted to see what the biological relevance of these differences were. And so I tested these introgressed females in the field. So we had females that were homozygous subflexa versus females that were heterozygous for this one chromosome, chromosome 20, that was introgressed from Heliothrus verrescens. And so we did these tests in the field where we tested one female in these sex pheromone traps so that we could see what type of males were attracted and how many males were attracted to these different types of females. And then afterwards, we would genotype and phenotype the females that we had out in the field. And so what we found is, first of all, that subflexa males were less attracted, not completely significantly, but they were less attracted to the females with these lower amounts of acetates. But what was more important is that the Heliothrus verrescens, so the other species, was now significantly attracted to these pheromones and to these females. So here we saw experimentally that acetates are really important actually to inhibit cross-attraction. So then, now that we had these two QTL, we could actually then map the candidate genes onto our genetic maps. And acetates are produced through acetyltransferases, and many acetyltransferases have been found in the genomes. And so we mapped these acetyltransferases, but they all mapped to different genomic regions. So none of them mapped to our QTL regions that we identified. So then Fred Gould mostly continued with this research to introgress this region to fine scale map. So at first in females there is no crossing over, but in males there is crossing over. So when you then, after a number of backcross generations, you can actually use the males again, and so you can make the QTL region smaller. And that turned out to become the family so-called DD23, where this one specific locus on chromosome 20 was now smaller and homozygous. So this is now a QTL region of 2.4 centimorgan, and this is where Arthur de Fouché as a postdoc and Elise Frutet as a PhD student came in, and they continued the research on this in this line. And what they found, first of all, is indeed that the relative percent of acetate esters was significantly higher in the homozygous SS females, how we call them, compared to the VS and the VV females. So that only were different in this one specific region. And then, of course, we could also see what kind of candidate genes were located in this QTL. And by now, Ron Guo and Megan Fritz at Maryland University have sequenced the genome of verrescens and subflexa, also with the help of David Heckel. And so they found that in this region, we have 4 candidate genes, namely lipases and esterases. And I just depict them here as an easy depiction of esterase 1 and 2. So in tandem they are, and lipase X and Z. What I'm showing is here the Heliothrus subflexa allele, and here the Heliothrus subflexa allele with this green introgressed verrescens bit. And so that region has 4 candidates, so to say. And to give you a better idea of how that works, or how we think that works, so we have here again, these alcohols are the precursors of the acetate esters. You would think that the transference or the conversion of alcohols to acetate esters is through acetyltransferases. But these acetate esters are also produced converted back to the alcohols through acetate esters and lipases. So then we found thus candidate genes that are involved in acetate degradation. So what we then try to see if indeed the fluorescence and the VV should have higher expression levels in these Canada genes, and that was indeed what Arthur found. So in the lipases, he found significantly higher expression levels, and the esterase also, but mostly it was the lipases that showed these significant differences. And I'm very happy to say, by the way, that we just heard that finally, after more than 20 years, all this research has been now accepted in Nature Communications. And so Elise, what she did is to see if this was really true. So she also did CRISPR on these genes. So she CRISPRed lipase X and Z and esterase 1 and 2, and also all the 4 genes, and she found the exact same result. And so indeed, as soon as they are knocked out, if the V allele is knocked out, then the acetate esters are back to the relative percentage of what you can find in the subflexa females. Okay, so that's great, but we had this other QTL still as well, so chromosome 28, and that also explained a significant amount of variation in these acetate esters. So there we try to do the same. We also try to integrate. So Fred tried this, David tried this, we tried this, and they all failed. So we always, it became lethal. So after a couple of generations, we found there were no survivors. And so we even tried, we thought inbreeding depression is very prominent in Lepidoptera. So we thought that maybe there is some lethality because we introgressed it so much. So they actually produced 2 different lines and then tried to cross it, but it always failed. So we could not do this for chromosome 28. But then David did find in this QTL region 3 esterases in tandem. And since we are now focusing mostly also on the degradation of these acetate esters, we thought, okay, that could also affect the acetate esters. And he found in one of those esterases, the CXE21, a transposable element. And so if our expectation is thus, if this transposable element disrupts this gene, then we should see more acetate esters in these females. However, when Elisa checked that, then in these females, where this CXE21 is just completely disrupted in subflexa, she found actually less acetates instead of more. And then she did CRISPR to see if that was still the case, and she CRISPRed this gene, and then she found the exact same results. So we found an opposite to expected effect, and we actually don't know what to make of this yet. But that brings me to the intraspecific variation in acetate esters in Heliother subflexa. So here I previously found that Heliother subflexa in the east coast of the US has a much higher level of acetates than on the west coast. And actually, I also found that to be a genetic difference. So when I brought them back into the lab, those differences remained, and so I could also do QTL analysis on these intraspecific variation. And interestingly enough, we found the exact same chromosome, chromosome 28, to be the significant difference, to underlie this variation. So here we see that the same QTL region, and it's a chromosome, but it is 1/33 of the whole genome. So the chances are not that high to find the same chromosome. So that's interesting. And so then we tried to find out what is then the underlying mechanism maybe in this variation between in Heliother subflexa between the east and the west coast. Well, one big difference in these 2 regions is that in the east coast, Heliother's fluorescence is omnipresent because that is a big pest in many different crops. While on the west coast, Heliother subflexa is mostly prominent. Let me see. Oh, sorry. And that is because, and actually I did a lot of research also on the West Coast, and I hardly ever found, I only found in the 2 months that I was there, at the end of the 2 months, I caught 3 fluorescence males out of the hundreds of subflexa males that I caught. And so that is because on the West Coast of Mexico, there is this big these big fields of physalis, that is the host plant of Heliothrus subflexa. And interestingly enough, Heliothrus virescens is a generalist and it feeds on many, many, many different plant species, but it cannot feed on physalis. And so here we have indeed a region where there is a non-overlapping area where Heliothrus virescens is not present. And so maybe if the acetate levels, high acetate levels are costly, there might be selection to reduce these acetate levels. And so that brought us to the question, are acetate levels or high acetate levels costly to maintain? And so here, Thomas Blankers and Emily Birdfield-Steele did selection experiments together with Elise in the lab. So over 10 generations, they selected for high acetate levels versus low acetate levels in Heliothrus subflexa, and they succeeded. So that was a significant increase in the high line, and the low line remained low overall, and it did not have an effect on the other pheromone components. So the only difference here is really the acetate levels. When we looked at, so Lotta Vaskimo was a student in the lab who looked at the expression levels of different genes, and she found indeed similar expression levels as what we found before. So lipases are higher expressed in the low line, which is similar to what we found before in Heliothrus virescens that don't have any acetates, while she found higher expression levels of the CXE21 in the high line, and that also is similar to what we found before, had this opposite to expected effect. And so then Elise mostly checked whether these acetate levels might be costly. And so here she checked, first of all, in the high line and the low line, how is the fecundity and the fertility affected by the pupal mass? So the pupal mass varies quite a bit in all these lines. And so when you see the low line doesn't have any, and there is not much variation, that doesn't matter, the fecundity and the fertility is not affected by the pupal mass. But in the high line, so in the high acetate producing line, there is a significant effect. So when the pupal mass is very low, then also the fecundity and the fertility is lower than in the low line. And only when the pupal mass is high, then the fecundity and the fertility is also higher. And so it seems like maybe there is indeed a cost of acetate production, although this is not convincing enough yet. And so she also did an experiment together with a student, Rick de Jong, where they reared the insects on different types of diets, so on normal diet, but also on diet that has reduced nutrition, and then checked also the acetate levels here in these different experiments. And here what she found is that in the individuals that were raised on the control diet, there was no effect. You don't see a change in the acetate level. But when they were reared on the reduced diet, then you see that there is indeed, first of all, that relative percentage of acetates is affected by the developmental time. So the developmental time is longer, then you see higher amounts of acetates, but also the pupal mass. When the pupal mass increases, you see a decrease in relative percentage of acetates. So it's not convincing all by itself, I think, but together I have the feeling that we are getting there. And then also I want to relate this to what I have found before, which is that when I first found these differences in acetates in the geographic regions, I was wondering whether this is a plastic trait also. So here we tested whether the pheromone variation changes when the females emerge in the odor of their own species versus the odor of the other species. And when they emerged in the odor of their own species, we didn't find any variation or difference in the acetate levels in the pheromone gland. However, when we had them emerged in the odor of the other species, we found a significant increase in the acetate levels. And so apparently females can adjust their pheromone depending on the chemical environment. And I think that this also suggests that acetates are costly. So to wrap up, I think there is, so it seems to me that the acetates are the ancestral states, having acetates in your pheromone gland, and that the species that do not produce acetates, that's the derived states, I think. And what I could imagine is that there is indeed levels, there's host differentiation, and so the acetate levels in subflexa depend on the level of communication interference. And when there is no communication interference anymore, there is a reduction in these acetate levels. And the absence of acetate esters in Heliothus virescens is maybe because the communication interference is minimal due to the population size. So they are so dominant that there is much less chance of attracting the wrong type of male in these regions. And to wrap up, I would like to show some final analysis that Elise did for her PhD, and that was this Canada gene that we now have, lipase X, and of the lipases. She checked in the genomes of a number of species that I see here show which ones have acetate levels and which one don't. And here we have the Heliothus subflexa and Heliothus virescens. And interestingly, there is a very similar pattern going on in Helicoverpa asulta and Helicoverpa armigera, which are species in Asia. There is a species that doesn't produce any acetates, that is armigera, which is the generalist, while Helicoverpa asulta is the specialist and does produce acetates. And these two species, it seems that there was a gene duplication happening, and then in the species that doesn't produce acetates, both of these genes are expressed, while one of this gene is not expressed in the acetate producing species. And so that is what we are then also expecting in this species pair. But that is something that remains to be studied. So with that, I would like to thank a lot of people, sorry, that were part of this long-term research that still remains to have a lot of questions. So with that, I would like to thank you.
Thank you very much, Astrid.
Shall I stop sharing?
Maybe we'll wait. So I think we can have one or two questions to Astrid now before we move on to the general discussion. So please, if you have questions, post them in the chat. And maybe I can start asking about, so in the sort of bigger picture, what is the role of the second QTL and the TE in sort of reproductive isolation in the system? If you could discuss a little bit about that.
Yeah, so we think that these two QTL interact with each other because, as you can see, each QTL, reduces the number of the amount of acetates, but it doesn't show any that you don't have any acetate esters, and so there must be some interaction going on. So that there is that you have a complete degradation of the acetate esters. Actually, it doesn't make any sense to me still that acetate esters are being produced first and then degraded again. If they are costly to produce, how would that work? And secondly, I would expect actually that in Heliothus virescens, so in the non-acetate producing species, I would expect that every now and then we would then just find some acetates. And I can easily say that I have extracted now at least 10,000 or more females and glands. And I have absolutely never found any acetate esters. But in Thiel and Tomlinson already in the 80s showed that if you put acetate esters on the pheromone gland on the outside of Heliothus virescens and subflexa, then the conversion towards alcohols is about 30 times higher in the non-acetate producing species, so in Heliothus virescens, than in Heliothus subflexa. So apparently, obviously, they do degrade acetates very, very quickly. So, and now with all these results, I think that I just have to get peace in, with the results that indeed there is acetate degradation and not a non-acetate production. I don't think I answered your question, but I don't have an answer to your question.
Yeah. Roger has a question.
Yes, thanks for the talk. You mentioned inhibition of cross-attraction. And I just wonder whether you think that's an evolved function or a chance side effect of divergence. Because if it's an evolved function, then it's a kind of clear example of interspecies interactions, kind of directing reproductive isolation.
Yeah. So did acetate esters evolve before or after they were in symmetry? Is that the question, maybe, or not?
Well, I think that's part of the question, is whether they evolve for the function of inhibition of cross attraction rather than evolve for other reasons.
That's a good question. It seems to me that since the acetate esters are reduced in regions where the heliophosphorescence are not present, that at least the high level of acetates evolved because of the communication interference. But that's my hypothesis. Is that answering your question?
Yes, I think partly. Yeah. Do you think that's common across moths in general, in pheromone systems in general, that they have things with inhibitory functions rather than attractive functions?
Well, I don't know. So there are indeed some examples, but those examples are not very many, actually. So you do see divergence in sympatric populations, so that the ratio changes, but the inhibitory function, I think, is not so commonly found. And I think that's an interesting aspect of these acetate esters. That I think you also see that the increasing in acetates esters, at least in the North Carolina population, increases also the attraction of conspecific males. But at the same time, it decreases the attraction of heterospecific males.
Yeah, thanks.
Yeah.
So maybe we stop the recording here.